All right, well, we had some problems with the projector, but we got those solved, as you see now. So, but let me go ahead and make a couple of announcements, and then we'll just jump right into materials. No riddle today, sorry, because I'm behind already, and I got to get this done. We're behind from Wednesday even. So, so in terms of announcements, hopefully you saw that I posted the exam scores yesterday. So, if you were to take the percent average of the combined scores between the true-false section and the short answer, it was a 73%. So pretty good, I think, especially considering the difficulty of the material. That regulation, carbohydrate regulation, is not easy to get through, for sure. So that kind of closes the book on that. I have put those exams along with your problem sets, problem set 2 in MRB 111, so you can pick them up. I also posted a key of that exam, so you can look at that. You should also be able to see your zip strip now, okay? So you've got the full amount of information. Please go ahead and look at it. If you have questions about that, please let me know, okay? I will say this, I think that, generally speaking, as I read through them, people struggle a little bit more with the glycogen metabolism regulation than anything else. So, and if you pick up your exam, you might find that's true of you as well, okay? Okay, so what we're going to do today, we're going to kind of shift gears now. We're going to shift into our second unit of the class, moving away, in some senses, away from carbohydrate metabolism into cellular respiration. So that would be our second unit, cellular respiration. And you see here just a kind of diagram of cellular respiration. We have come to this point essentially in carbohydrate metabolism, and that is that carbs feed into this molecule pyruvate. That molecule, as we're going to look at in chapter 7, or lecture 7, is going to be converted to acetyl-CoA. That's what we're going to do essentially today. I hope to get it done by today, so that we do not have to come back on Monday and Monday after break and discuss it. My plan for the Monday after break is to do another group work assignment on the citric acid cycle. Okay, so that's the plan, and I'm going to try and stick to that, hopefully, regardless of where we get today. Okay, so but pyruvate will be converted to acetyl-CoA. Now, the other aspect of this that I want to go ahead and point out here for us, and that is that this again feeds into this larger context, because we can take amino acids or we can look at fatty acids, and those also will feed into this molecule acetyl-CoA. Okay. Amino acids are a little bit tricky because they can also feed into glycolysis or pyruvate or into the citric acid cycle. Okay, so we'll look at that more closely as we get into the later stages of the class, in particular amino acid catabolism. All right, but nonetheless, there's this central metabolic intermediate, that's acetyl-CoA, okay, that these catabolic pathways will feed into. Now, acetyl-CoA then will feed directly into the citric acid cycle. Okay, the citric acid cycle will be covered in lecture 8. Okay. Now, that pathway, just like glycolysis, can produce a small amount of ATP through substrate level phosphorylation. Okay, glycolysis, as we've discussed, of course, in this class, can also lead to ATP through substrate level phosphorylation. The citric acid cycle will also, though, and this is really important, produce significant amounts of reduced electron carriers, NADH and FADH2. Right? These other processes, as we saw like with glycolysis, can produce reduced electron carriers. In particular, fatty acid metabolism, that can produce a lot of NADH, a lot of FADH2. We call these reduced electron carriers, or carrying electrons from these carbons in these oxidative pathways. Okay? And we, of course, talked about here already these shuttles that get those molecules, in particular NADH, into the mitochondria. Okay? But the last thing that we're going to look at here in cellular respiration then would be taking all the electrons here, 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 and here, and shuttling those into what's called the electron transport chain. And by and large, oxidative phosphorylation, which is another way to make ATP. You have to have oxygen to do it, though. Okay, and that will be covered then, of course, after the break, lecture 9. Okay. Now, as you look at this, honestly, you could consider cellular respiration as occurring in 3 different phases. Okay. The first one being essentially the production of acetyl-CoA molecule through these major metabolic routes: glycolysis, amino acid catabolism, fatty acid catabolism. Okay, that's the production of acetyl-CoA. That would be the first major phase of cellular respiration. Second major phase of cellular respiration is the citric acid cycle, the Krebs cycle. Okay, central metabolic pathway really of every living organism. And then finally, that last phase would be oxidative phosphorylation. Okay. We're going to look at, of course, all 3 of those. We've already gone part of the way in looking at the first one in terms of landing at pyruvate through glycolysis. In fact, okay, what we're going to be doing today is looking at how we can take the next step from pyruvate to acetyl-CoA. Okay, and I wish it was just an easy thing to do, just draw an arrow and we'd be done. It's not quite that easy. Okay, we're going to look at this enzyme, pyruvate dehydrogenase complex, okay, in today's lecture. All right, we'll first think about, in terms of the enzyme, just it being this complex. It has this really complicated architecture. We'll look at that. Okay. After that, we'll consider the mechanism, and then finally the regulation. Okay. I'm going to move through this pretty quickly. I don't want to spend a ton of time on this, but I do want to give you some exposure to this. Okay. Beyond just saying, and pyruvate becomes acetyl-CoA. Next. Okay. All right. Now let's do this. As a reminder, we got sucked into all these kinases and glycogen metabolism and all that kind of stuff. So let's just come back here and start with glucose, give you the big picture again, okay? Come to pyruvate, okay? Of course, you need NAD plus to do this, as we've discussed several times, and you're going to get some NADH from this, okay? And of course, this leads to ATP production as well, okay? So that's everything that we've talked about in the cytosol, okay? Now, what we can do is, in terms of expanding this, we have talked about this as well, and that is pyruvate can become lactate. Okay. We talked about it as recycling the NADH in the cytosol. That's one of the things that can happen to pyruvate. Okay, so this would be essentially a redox reaction that we looked at. Okay. Something that I've mentioned briefly that we'll certainly talk about more thoroughly as well, and that would be the fact that alanine, an amino acid, is a single step away from becoming pyruvate. Okay, through a transamination reaction. A fairly important one. We will talk about that certainly more when we get to amino acid metabolism. For now, I think it's important to recognize that both lactate and alanine, or other amino acids, okay, are sources of pyruvate in the cell. Certainly glucose is. Other carbohydrates are, because they can feed into glycolysis. But lactate is an important source of pyruvate. Alanine certainly is. We could put other amino acids here. They'd take more steps to become pyruvate, but we could put other amino acids. Okay, so I could put this amino acids, step, 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 step. Okay, some more, some less, depends on the amino acid. Okay. So there's quite a bit of metabolism that feeds into the molecule pyruvate. Okay. Again, to try and expand us beyond just carbohydrate metabolism. Now, that then brings us, okay, into the mitochondria. And we're moving for the next 3 lectures, lecture 7, 8, and 9, we're going to move right into the mitochondria. Okay, pyruvate is going to be shipped into that environment. We don't have to cover a shuttle. You can be grateful for that. There is a symporter. It's actually the pyruvate proton symporter. Okay, it moves a proton as well as pyruvate directly into and through the inner membrane of the mitochondria. So the pyruvate comes here. Okay? Now, pyruvate can then undergo, and this is what we're going to do today, can undergo an oxidative decarboxylation reaction to become acetyl-CoA. Okay? Okay? That is today. Okay? And that's oxidative decarboxylation. Okay? Now, there's one other thing that I want to go ahead and mention now because it will pay dividends later on, okay, that we will discuss. We've actually already looked at this, but this is going to be an important aspect of mitochondrial metabolism, in particular for the citric acid cycle, not as pyruvate can become this. It can become 2-oxaloacetate, or oxaloacetate. Okay? And that would be pyruvate carboxylase that does that. That was one of the enzymes, in fact, that was the first enzyme in the gluconeogenic pathway. We put it in that context, rightly so, okay? But we're going to put it in other contexts as well moving forward, okay? It's an important reaction beyond just gluconeogenesis, so this would be carboxylation. Okay. You need HCO3 to do this, and some ATP as well. Okay. So if you start to look at this, right, there are several sources of pyruvate, several fates of pyruvate. Okay. Sources, certainly any carbohydrate, many of the amino acids, lactate. Okay. Fates of pyruvate, anabolically, we could go back in the opposite direction, right? Carbohydrates, amino acids, or lactate. Okay, pyruvate can also come in here. It can be converted to acetyl-CoA, and then pyruvate can become oxaloacetate. Now, we'll extend beyond this. Acetyl-CoA can become fat. So it can come back out here, and we'll talk about pathways that will make fatty acids from this. Or cholesterol, and from cholesterol, hormones. Okay, so there's a lot that can come from this molecule, pyruvate, or acetyl-CoA. Okay, we can make fatty acids and cholesterol from it as well. Okay, that's the big picture. Questions on that? Big picture for the next, couple weeks, and also for even today. Awesome. Okay, now, we want to do the stuff in the red box. So let's forward. We're going to not do that question here. Let's go forward here and let's look at this. Okay? So we could just go off to spring break right after this slide. There you go. Done. And you say, oh, awesome. That was great. Okay? Some of you are saying, let's just do that. I'm okay with that. It's okay. Some of you are saying, where's everyone else? I don't know. You're like the 33% of you. I actually counted you. I had time because I was waiting on this. There's 30 of you here. There's 90 in the class, so good job. You should get a little, anyway. Some of you are saying, yeah, I should get something, but you're not going to give it to me. Okay, I'm sorry. But anyway, all right, so this is our reaction. Pyruvate dehydrogenase complex is the enzyme that's going to do that. Now, this is complicated. It's a lot more complicated than really anything that we've seen at this point in time in terms of conversion of something to something, okay? To get pyruvate into, or to be converted to acetyl-CoA, it actually requires 3 different enzymatic reactions, okay? We have seen and observed scientifically that those 3 enzymatic reactions are found in this molecule called pyruvate dehydrogenase complex. Question, yes?
Is pyruvate dehydrogenase complex only found inside the mitochondria?
Yes. Yes. So the question that was asked, and it's an important one, is this found only in the mitochondria? Yes, it is. Okay. So, I mean, in terms of eukaryotic metabolism and mammalian metabolism, we move right into the mitochondria at this point. Yes. Okay. Good question. All right. So this pyruvate dehydrogenase complex, sometimes it will be called PDC. You'll see that abbreviation certainly in the literature. Others will abbreviate it PDH complex. Those are fine. Okay. As I mentioned, it's really 3 enzymes in one, and those enzymes go by these names E1, E2, and E3. We'll look at the architecture of those here in just a moment. The enzyme also, as you look at this, requires 5 different coenzymes. Okay? So this is a fairly major process in the cell. All right? That requires a significant source of coenzymes and a really large enzyme complex. Okay. That's going to make acetyl-CoA. You will also notice here really quickly and importantly, this makes CO2. Okay, we are removing one of the carbons from pyruvate. This is why we call it oxidative decarboxylation, because a CO2 is removed. Okay. That CO2, generally speaking, will become bicarbonate in the cell. It will also make NADH. And that NADH can go on and do things. In the context of catabolism, it will go on to make ATP. Delta G of this reaction, before we get off of this slide, actual conditions, delta G is less than 0. This is an exergonic process. It is irreversible and therefore regulated. In fact, it's tightly regulated. We'll hopefully have a little bit of time at the end of the day, or if we have to, after the break, to look at the regulation of this. Okay. Get one slide on that. Now, let's do this. Okay, and of course it makes acetyl-CoA. I want to go ahead and look at acetyl-CoA first, so you have an idea of the molecule that's being made. And then I want to go ahead and move on to the enzyme and the mechanism. So let's look at first acetyl-CoA. Here, you think, here is acetyl-CoA. Okay, that's how acetyl-CoA is typically drawn. The acetyl group, right, these carbons, those come from pyruvate. Okay, coenzyme A is this thing here. This, well, it's in this entire thing. Okay. You don't have to memorize the structure of that, but that's coenzyme A. Okay, some of you are saying thank you. Actually, here's what we'll do. The 66% of the people that aren't here have to memorize that. You don't have to. Okay. All right. I have no idea how I'm going to do that, but we'll figure it out. Okay. But no, this is the structure of coenzyme A. What's shown here and highlighted in your book, this actually comes from vitamin B5. Okay. Of course, the other portion here, this here is an adenosine nucleotide, ADP, and then finally there's this, on the other end, there's this mercaptoethylamine, okay? If you look at this, let me go ahead and erase these things, okay? Look at this arrangement here, that is a thioester. And it's a great way to store energy. Okay, the arrangement of bonds stores energy. Okay. We will encounter coenzyme A in other contexts. Okay. Certainly here it's connected to this 2-carbon acetyl group. Okay, that's our context now. Pretty much every fatty acid that would be found in the cell, okay, is going to be attached to a coenzyme A molecule. It's very rare to find just free fatty acids in a cellular environment. They usually are attached to a coenzyme A, and they're called then acyl-CoAs, not acetyl-CoAs, acyl-CoAs. Okay, and we'll talk about those, of course, during fatty acid metabolism. Now, on to then the enzyme. Okay, questions on anything that we've talked about thus far? Okay, great. Let's talk about the enzyme, okay, the enzyme complex. Right, so as I mentioned, pyruvate dehydrogenase complex is 3 enzymes in one, okay? And I mentioned on that last slide coming up here, I said these enzymes we can call E1, E2, and E3. Okay, we can and we will call them that, but they also go by other names. So let me go ahead and label these. This is E1, also called pyruvate dehydrogenase. That would be subunit 1 of pyruvate dehydrogenase complex. Subunit 2, this is a fun name to say and spell. This is why I like E2. It's dihydrolipoyl transacetylase. Okay, so that's fun. And just like it, dihydrolipoyl dehydrogenase is E3. Okay. Now, to give you some sense of this, okay, let's say copy number here. And then we'll say coenzyme here. Make kind of a table here. Pyruvate dehydrogenase complex has, the ones that we've observed in mammalian cells have 60 copies of E1. That's a large number of subunits. Okay. Furthermore, they have 60 copies of the transacetylase. Now we're at 120 subunits in a single enzyme. Okay. And then finally, and this is strange, there's 12 of these. Okay. Okay. But there's actually, those are trimers, so there's actually more than that. But anyway, there's 12 of those. Okay. So a total of 132 subunits. Right. Huge enzyme complex. It can be seen with microscopes. Okay. Very big. Okay. Electron microscopes are actually the way that we have visualized this enzyme up to this point in time. Okay. In terms of the coenzymes that I mentioned, the pyruvate dehydrogenase has TPP in it. Okay. TPP is thiamine pyrophosphate. Okay, that's what TPP stands for. Okay. E2 will have the coenzymes lipoate, and also this is where coenzyme A binds. Okay. Of course, coenzyme A is going to be attached to the acetyl group and going to leave with that, so it's not a permanent association with it. And then finally, down here, the dehydrogenase has the coenzyme FAD, and also NAD will bind to it and release as NADH. Okay, so those are your coenzymes and where they're located in this complex of enzymes. Function-wise, pyruvate dehydrogenase decarboxylates. This will help us later on. Pyruvate. Okay? It will also carry out a redox reaction. Okay? So those are the two things that we will observe as we move forward. Decarboxylation of pyruvate and then a redox reaction. The transacetylase is going to make acetyl-CoA. Okay? And then finally, dihydrolipoyl dehydrogenase is going to make NADH. Okay? So in terms of tasks that need to be done to decarboxylate pyruvate, this is how they're set apart. Pyruvate dehydrogenase decarboxylates pyruvate, carries out a redox reaction. The transacetylase makes acetyl-CoA, and then finally the last dehydrogenase makes NADH. Okay. There's lots of details in that, but that's the overarching scheme. Question?
Would there be a coenzyme for each copy?
Yes. Or multiple.
Times 60.
Yep. Yeah, so the question that was asked, is there a coenzyme for each copy? Yes, there are. Okay, because each of these copies is carrying out these functions that I'm stating, and they will need the coenzymes to do it. So in one enzyme, it's not just, in one enzyme complex, there will be multiple reactions going on at the same time. Okay. Somewhere around 60, up to about 60 reactions at a time. Yes? So that, I don't know the answer to that. I mean, that's the way we observe them. The complex is, if you think about it this way, when we observe the complex, we take it out of its natural environment, and it's still associated as a complex. Okay, meaning that quaternary structure that we're observing is quite stable. So it's unlikely that you will find many of these enzymes operating as like lone wolves. I'm sure that we know, I just don't know. I don't know if you can take and separate E1 from the rest of them and have it function. Here's what I would say. It will function to a point, okay, but it will stop. Like for instance, I can say this, pyruvate dehydrogenase alone could decarboxylate pyruvate but could not carry out the redox reaction because it needs a coenzyme from E2 to carry out the redox reaction. Okay. So they can do parts of the function, but they couldn't do the full thing. Yeah. Other questions? And I'll kind of expand on that answer a little bit because we're going to see how one of these coenzymes actually carries substrates, some of the substrate around to the other active sites. Okay. Other questions? Let's do this. Here we go. The soccer ball. Okay. That's pyruvate dehydrogenase complex. If you have a super powerful microscope, it looks red, green, and yellow. Okay. All right, not really, but anyway. Okay, so what you're looking at here, they've taken apart the molecule a little bit by a little bit, but what you are looking at, what I just circled, that would be essentially. Let me see here. That's E2. Okay. Now, truly, this does look like a soccer ball, right? I mean, you go over here and they put these little red things in it, and it looks like a soccer ball. All right. That red that you're looking at, that is E3. Okay. And then if you come down below here, right? Surrounding the soccer ball is another ball, okay? A ball of yellow. And the ball of yellow is E1, okay? All the E1 subunits. I think D is probably the best way to look at this, panel D here, because what they've done is they've essentially taken C and cut it like an apple, cut it right down the center of it. And so you see then here E1s, you see here the E2s, and you see here finally the E3s, and their arrangement with one another. Now, this I believe is the structure from like either yeast or E. coli. There's less subunits in those organisms. It's a smaller molecule. Okay, but nonetheless, this is what we'd expect mammalian to look like as well. Okay. Now, coming out of, and this will help us here in a little bit, but coming out of E2, there's going to be this arm. And I'll just draw it like this. Okay? That's really hard to see. Some of you are saying, I can't see that whatsoever. Okay, well, let's do this. We'll do it like this. Okay? It's not quite right. I have to do one more thing here. Sorry. I'll get there. There we go. That's better. Okay. And I'm just going to call this an arm. Okay. And it extends from E2. It's actually what will be called lipolysyl arm. It's this 14 angstrom extension that comes out of E2. And it literally can swing around to each of these subunits. And it will carry either electrons or carbons from one subunit to another. Okay? That's a really poor rendering of this, but that's what we would see. Okay? And coming out of each of them, the E2s, is this arm. Okay? Now what we are going to do in future slides, we're not going to look at the whole complex. We're going to isolate 3 of them, and we're going to show you what happens. All right, but that arm is going to play a really important role in our understanding of this. Okay, but the arm carries, let me say this, carries electrons, and it'll really carry an acetyl group. Okay, I said carbons, but it's really more accurately acetyl group. Questions? Yeah.
Do we know if it's different steps of the reactions that it's going to be happening all different times, or is it like one big concerted division?
Yeah, so the question is, do we know if all different steps of the reaction are occurring at a time? So in different regions of the pyruvate dehydrogenase complex, it would be reasonable to think that they would be doing it at different times. In fact, given that you only have 3 E3 subunits, you'd expect them to be doing it at different times. Okay. Whether there's like some, right, whether they're synchronized and that kind of thing is, I don't think we know. I think it would be really almost impossible to do that, at least with our experimental technology now. But the expectation is, yes, they would be different subunits would be doing different things at a time. Yeah. Other questions? Okay. All right, let's do this. So coloring scheme is important. Green, red, and yellow. Green is E2, E3 is red, and E1 is yellow. Okay, so we have here then, okay, essentially the 3 subunits, and you can see then the labels here. E1, and then green is E2, and then finally red is E3. Okay, we are going to do this. We're going to be kind of looking at this from a surface level because we had class was canceled on Wednesday. Okay, which was okay, I'm sure, with all of you. So we're going to look at 5 different steps of this enzyme. Okay, using this figure. Okay, and the very first step is going to be this. Step number 1. Okay, and look at this. Number 1, this is decarboxylation. Okay, pyruvate's going to enter into this. You're going to see CO2 leave, and you're going to get essentially the acetyl group. Okay, that acetyl group is going to be attached to TPP. Okay, you have to have TPP to do this, thiamine pyrophosphate. Okay, it is a coenzyme that's absolutely required to decarboxylate pyruvate. It produces a TTP hydroxyethyl intermediate. Okay, hydroxyethyl is 2 carbons with the hydroxyl group on it attached to TPP. That's where our nomenclature comes from. TPT hydroxyethyl intermediate. Okay. And that would be step 1, okay? TPP hydroxyethyl. Is produced. Okay. It's a covalent bond between the hydroxyethyl and the TPP. Now, if you are super interested in this, and this is what I mean, we're going to glaze over this. If we had come to class on Wednesday, we might have gone through this slide. And you sit there and say to yourself, I'm really upset that we didn't get to come on class on Wednesday, because that looks like a lot of fun organic chemistry. Okay, so you can do that on your own time. It does. I mean, it's important to think about these things as happening more than just arrows, right? I mean, this is the organic chemistry behind what I just said, okay? You end up with this hydroxyethyl intermediate, okay? Now, the next step, coming back to this, step number 2, this guy right here, I'm going to circle this here, come all the way up here, like this. That step, number 2, is a redox reaction. Okay? And you'll notice here, I'm going to put here arm. Because the arm is what's going to be important. Okay? The arm is extending out of E2, extends into E1's active site, and it is going to be part of this redox reaction in which these carbons from the hydroxyethyl are going to be transferred to the arm, and you'll see here this guy right here. Okay? The bond angle on that looks a little fishy, but nonetheless it's going to make this, it's going to attach the acetyl group to that arm. Okay? So here's our arm. And we could say that this is the reduced arm. And this is the oxidized arm. Okay. That's step number 2, redox reaction. Question?
Maybe it's not clear, but does the arm itself swing, or does the whole subunit?
The arm itself is moving. Yep, so the question is, does the arm itself move, or is it E2 that's moving? It's the arm. Okay? Yep. And this is not unprecedented in other, I mean, there's other enzymes that have these arms that reach into things and then carry things to other subunits. Okay, in fact, we looked at pyruvate carboxylase that makes OAA. That has an arm that has biotin attached to it. Okay. So anyway, but let's go ahead and look at this arm. Let me come here. So this is lipoic acid. Okay, if we're actually, this is not lipoic acid. I don't know why it's labeled that way. It's lipoate, the way it's drawn. Okay, because they've got it deprotonated. Lipoic acid would have put a proton in that group. That is a coenzyme. Okay, now that's lipoate. So the arm is actually this lipolysyl group that I keep calling the arm. Okay, here is lipoate. It's essentially a fatty acid coenzyme. That is covalently bound to a lysine side chain. And then this, all of this, here would be E2. Okay? The whole thing is 14 angstroms long. It's really long and it's really flexible. Okay? It can reach into other things. It can move all around. Lysine is a very flexible amino acid. The lipoate is a very flexible coenzyme. Okay? Now, this is the oxidized form. We look here at these sulfhydryl groups. That's oxidized. Okay? The reduced form, I don't have room to draw the reduced form, but let me go ahead and draw it up here, would be with SH. Right? R. That would be reduced. Okay? So that's your arm. Coming back to this then, right? We have up here, I said the reduced arm up here because it's got that acetyl group on it, but it's the thiol groups have been reduced. Okay? It's carrying electrons, it's carrying an acetyl group, going back to some of the things that I've said it would do. Okay? Now, you say, some of you are wondering, okay, there's that wonderful arrow there, like magic. Magic. It's not really magic, it's this. If we were on class on Monday or Wednesday, we would be talking about this. Okay, we're not going to do that. Okay, actually it would be part 1 that we were just looking at. Okay, the magic that I was just referring to is this magic up here. Okay, on my next slide, okay, step 3, let's come back up here. This next portion here now happens in E2. Here we go. This is, right, step number 3 makes acetyl-CoA. Okay? The enzyme's name is trans-acetylase, so this happens in E2. Right? The arm reaches into E1, grabs the carbon, the acetyl group, gets electrons, carries the acetyl group into its own active site. Coenzyme A is waiting there, and it makes acetyl-CoA in step 3. The enzyme name E2 is trans-acetylase, meaning it's taking an acetyl group off of the arm and putting it onto coenzyme A. Okay, it's just moving an acetyl group, trans-acetylase. Right. Acetyl-CoA is the production of this. Right. Now, you have at the end of that, you'll notice here, still the reduced arm. Okay? What happens here? Magic. This magic. Okay? That we're not going to talk about. Yep.
I thought you called the third arm the oxidized arm.
Yeah, okay. Coming back here, away from my magic. Okay, the third arm. So you said, I thought I called the third arm an oxidized arm. This would be the oxidized arm. That's the reduced arm with, it's really an acetylated reduced arm. Maybe I should be more clear in this. It's acetylated. Okay, reduced arm. It loses its acetyl group, becomes the fully just thiol containing group. Okay? And those then are going to pass electrons on to NADH ultimately. Okay? Now again, we're glazing over lots of details here, and you say to yourself, that's just enough detail. I mean, what you're doing is great. That's the right amount of detail for now. Thank you before spring break. I'm sure that I'll go contemplate these things throughout my break. Okay. The people that didn't show up today are going to have to contemplate this at some point in time. Okay. All right, now, down here, step 4 and 5, I will put on a single slide. Let's do this. Let's find some empty space here. Steps 4 and 5. Okay? Steps 4 and 5, I guess I should come back here. I'll come back here and just highlight this. This guy right here, number 4 / number 5, make NADH. Okay? That's steps 4 and 5. Now, the figure is technically correct in what it does. There's some even more detail than I would want to go into on that figure. So what we'll do here is this. We'll just draw it this way. Okay? So we have our SH groups. Okay? This is connected to E2. I'll just put R here. This is going to give its electrons and become this. So again, this is on our arm. Okay? Those are going to give electrons to FAD, making FADH2. FADH2 is going to give its electrons to NAD+, making NADH plus a proton. This will go on to electron transport chain. Okay. I don't even have a slide to show this magic, okay? I wasn't ever even planning on doing that. Okay? But this would be an okay way of doing this. This all happens in E3. Okay? And then the whole cycle starts again. Okay? So, to summarize, okay, obviously that's a duplicate of this slide. There are 5 steps to get pyruvate, about 5 steps, 5 overarching steps to get pyruvate to become acetyl-CoA and also pass its electrons to NADH. Okay. First step is a decarboxylation step. Second step is a redox step to make an acetylated reduced arm. Happens in E1. The acetylated reduced arm goes to E2, and that's where transacetylation happens. An acetyl group is passed to coenzyme A. The arm remains in the reduced form, swings into E3, passes these electrons to FAD, and then to NADH. Okay. Step 5, and then you're back to this. Okay. This is happening, okay, in all of these complexes. This is kind of like the question that was asked earlier. Are they happening at different times? You'd expect that E3, because of the number of E3s, is going to be accepting electrons from different arms. Okay. Going back to the question, can they operate separately? They could certainly do some of these things separately, but not in totality. Okay. The arm is kind of crucial for E3 to work at all. Okay. All right. And that brings us to spring break. Okay. So we will come back and we'll do this slide probably the Wednesday after spring break. Monday after spring break, we'll jump right into a group work assignment on the citric acid cycle. Make sure you're here on Wednesday to get credit, or not on Wednesday, but on Monday to get credit for that, okay? All right, have a good break. I know that we all need it at this point.